Hello everyone. So today we are going to discuss our topic called PCR, Polymerase Chain Reaction. Now PCR, it's a molecular technique and we use this technique to amplify a specific stretch of DNA. Now when we perform this technique in any molecular biology lab, we have to make a reaction mixture where the PCR reaction will take place. Now in the reaction mixture, we actually mix different type of component starting from the DNA template. Now in the DNA template, this is the template which we use to amplify our specific or interested DNA. Now the second thing is the primer. Primer is a single stranded small DNA molecule which used to perform the PCR reaction. Now in the PCR reaction, we generally use two different type of primer. One is called the forward primer and another one is the reverse primer. Now how the forward and reverse primer will work, we will going to discuss this later. Now this reaction will take place in a reaction buffer because this buffer stabilizes the pH and it also protects the enzyme which we are going to use to perform the PCR reaction. Now the buffer, it must contain some metal ions, for example magnesium. Along with that, we need the DNTP. Now this DNTP is the building block in the PCR reaction. As I have already mentioned that during the PCR reaction, we use this technique to amplify the genomic, to amplify the stretch of DNA. Now when we try to amplify a DNA, that means we are going to synthesize multiple copies of a specific stretch of DNA. And for that reason, we need the different type of DNTPs. Now this DNTPs includes all the four type of DNTPs including ATP, CTP, GTP and the CTP. And along with all those things, we have to add a polymerase. That's why the name is polymerase chain reaction. Now this polymerase is the vital component in this PCR reaction. Now there are multiple type of polymerase are there as we know, and this polymerase help in DNA replication. Now in the PCR reaction, we have to use a very special type of polymerase which must be heat stable. The normal polymerase which present in our body or in prokaryotic or eukaryotic cell, most of them they are thermolabile. They will not able to sustain at a very high temperature. But in the PCR reaction, we have to use a special type of polymerase which is heat stable. The most commonly used polymerase is commonly known as Taq polymerase, as the polymerase was isolated from Thermus aquaticus. It was a bacteria which can survive even at the temperature of 100 degrees centigrade. So that's why the Taq polymerase is a special type of polymerase which we use during the PCR reaction. Now in the PCR reaction, the reaction actually happen in multiple steps which I am going to discuss right now. Now we are going to discuss the steps of PCR. Now the PCR reaction is contain 5 different stages. First is the initial denaturation, then denaturation, annealing, extension and then final extension. Now what happened in these steps? Now in the denaturation step, or most precisely in the initial denaturation step, we increase the temperature at 95 degrees centigrade and we maintain at the 95 degrees centigrade temperature for at least 2 to 3 minutes. Now in this high temperature, your entire DNA strand, it will get separated. The double stranded DNA strand, suppose this is the double stranded DNA molecule. Now in the denaturation step, due to this high temperature, this double stranded DNA strand, they got separated and it's become single stranded. Now after that, we comes to the annealing temperature. Now annealing temperature is here the temperature is a little bit variable. It can be as low as 50 degrees centigrade or it can be as high as 60 or sometimes even 65 degrees centigrade. Now this temperature is actually depends on the primer. As I have already mentioned that during this PCR reaction, we need the primer sequence. Now there was two type of primer sequence. One was the forward primer, another one was the reverse primer. Now when we try to amplify the DNA strand, as we know that the DNA cannot be synthesized de novo. That means when the DNA strand is separated, even if we add the DNA polymerase and the DNTP, the DNA polymerase will not able to synthesize a new strand of DNA because DNA cannot be synthesized de novo. So what we have to do, we need to provide a small single stranded DNA molecule. A pre-existing strand of DNA is required for the DNA synthesis. So this pre-existing DNA strand is called primer. Now primer, they are single stranded DNA molecule. They are single stranded DNA molecule and they are very small in size. Generally the size of the primer varies from 20 base pair to up to 30 base pair, and they are very unique. They will only bind with the DNA at a specific location. So Manu, this is my DNA strand when they got separated, and when we reach at the annealing temperature, this primer, it will bind with the DNA strand, and this small single stranded DNA fragment or the primer, now it's act as a pre-existing strand. Now the 3' end of the primer, it will be recognized by the polymerase. The polymerase will bind here and then it extend the strand. So this is the molecular basis how a polymerase can synthesize the DNA. So the same thing happened in case of PCR reaction. So in the PCR reaction, in the initial denaturation step or in the denaturation step, we increase the temperature so that the double-stranded DNA converted into the single-stranded DNA. Now in the annealing temperature, we actually allow the primers to bind with the DNA molecule. So in the annealing temperature, we suddenly decrease the temperature from 95 degrees centigrade to around 50 to 60 degrees centigrade. Now when we decrease the temperature, these two single-stranded DNA, they try to anneal again. But as the DNA size is much more longer in compared to the primers are small molecules, so they will easily able to anneal. So when we decrease the temperature and when the temperature reach around 50 or 60 degrees centigrade, immediately the forward primer and the reverse primer, they will bind with the single-stranded DNA. The forward primer is actually have the same sequence of the same strand, which is 5' to 3' DNA strand, and the reverse primer, it has a reverse complementary sequence of the gene. So that's why it will bind with the same strand, and the forward primer, it will bind with the antisense strand. Now after that, we have to increase the temperature, and this step is called extension, where the temperature is 72 degrees centigrade. Now this temperature is the optimum temperature for the activity of the Taq polymerase. So at that temperature, the Taq will start synthesizing the DNA, start the DNA synthesis process, because in the annealing temperature, the primer already attached at their specific area or the specific site. After that, the polymerase, they will recognize this joining, because this is the orientation of the strand 5' to 3', and when the primer will attach, this blue color is denoting the primer. This is my reverse primer and this is my forward primer. Now when the reverse and forward primer bind, it will give a free 3' hydroxyl group, and this 3' end will be recognized by our Taq polymerase. It will bind here and it will extend the strand. So from a double-stranded DNA, now we have two molecules of double-strand DNA. So basically we have synthesized one new copy of the DNA. Now we are going to repeat the process again and again. That's why the name is polymerase chain reaction. So we convert this reaction into a chain reaction. Now the reaction will goes on. How many times? Generally we prefer 32 cycles. Sometimes we can extend it up to 40 cycles, depending upon the amount of the initial DNA we use. So in the denaturation step, the DNA will become single stranded. Then in the annealing step, the primer, it will bind with the single stranded DNA, and then in the extension stage, the polymerase will recognize the DNA and primer hybrids, and then they extend the 3' end and resulting to the amplification of that particular DNA. And after that, we don't need this initial denaturation and the final extension every time. Just at the initial stage of the PCR, we need the initial denaturation. After that, only the denaturation, annealing and extension. These three steps will repeat again and again and again. So this is my initial denaturation, and this is the cyclic step. Denaturation, annealing, extension. Again denaturation, annealing, extension. Again denaturation, annealing, extension, and it will goes on. And after the finish of 32 cycle, we will give a final extension. This final extension actually provides some extra time to the polymerase so that they are able to synthesize if there are anything incomplete or half done. So we just provide some extra time so that the polymerase, they are able to finish those process. And during the PCR reaction, some polymerase, for example Taq polymerase, they have a special features that after the synthesis they generally add one extra A at the 3' end, and that extra A help us to perform some type of molecular cloning, for example TA cloning. So in the final extension we also allow the polymerase to add that extra A in the 3' end as well. So this is the utility of maintaining an final extension. But in a normal PCR cycle, this final extension and this initial denaturation, it will take place only once. But in between this denaturation, annealing and extension step, this step we have to perform multiple times. That's why we call it polymerase chain reaction. Now here, during this PCR reaction, the amplification is actually they follow the geometric amplification, the GP. Because if the number of DNA strand is 1 in the first cycle, suppose we start from only 1 molecule of DNA. Now after the first cycle, this 1 molecule it's become 2. And now in the next cycle, from 2 it's become 4. And this thing goes on. From 4 it will become 8, then 16, then 32, then 64, 128. So in short, we can represent that the number of amplified product is 2 to the power n, where the n is the number of cycle. Now we are going to discuss the application of PCR reaction. Now there are multiple areas where we can apply the PCR technique. The first is the gene cloning. So PCR technique is a molecular technique which we generally use in the molecular biology lab, where the PCR technique is mostly used for the gene cloning purpose, because we have to identify multiple genes and we generally try to clone that gene so that we can work on that particular gene. We can transfer that gene in order to make some genetically modified organism, or we can analyze the expression of the certain gene. So in every case, first we have to clone that particular gene, and for that cloning we have to amplify the gene from either from the cDNA or from the genomic DNA, and PCR can be used in this purpose to clone the desired genetic material. Then in the diagnostic, because in the diagnostic we can use the PCR technique to identify for the identification of multiple disease. Mostly the viral disease. During the viral disease, the viral DNA is actually present incorporated in our body. So in that case, we can take help of the PCR reaction to amplify the viral DNA, and the positive result shows that the person is actually infected by that particular virus. So in the diagnosis, in the identification of a disease, the PCR technique can be used. Then is the forensic science. In the forensic science, generally we can amplify the DNA fragment from very small sample, for example blood sample or from hair sample, that can help us to identify the suspect. Then in the agricultural as well, in the agricultural field we can also able to use the PCR technique, for example for the identification of genetically modified organism. Because in the genetically modified organism, mostly we actually incorporate some foreign gene inside that particular organism. So use the PCR technique, we are able to amplify and we can analyze whether that particular crop is contain any foreign gene or not. And last but not the least, we can also use the PCR technique for the identification of organism. Mostly in case of bacteria or in case of microorganism, the identification is a challenge. In that case, we can use the PCR technique to amplify a certain stretch of DNA, and after performing the DNA sequencing, we are able to identify the particular bacteria or that particular microorganism. So that's all about the PCR reaction in short. Thank you.